Hi and welcome to the kickoff of the Luminocean webinar series on marine biology. Our aim with this webinar is to present marine biology and conservation topics in a way that everyone can understand. So it's not just meant for scientists. It's specifically targeted for anyone who enjoys the underwater world, loves diving, or has an interest for marine biology. My name is Mareike Huhn, and I'm going to share the first few sessions, and I hope that in some of the next sessions after that, we will have different speakers here. We happily invite everyone who has a good topic, interesting topic to share. So please, sit back, relax, and enjoy our webinar. Shall we start?
Yeah.
People can come in as we go.
All right. Okay, so I will do the introduction first, and then after my introduction, we will continue with Mareike's webinar. Okay, good morning, Europe, and good evening, Banda. I myself now is in Kuala Lumpur, Malaysia, so it's 1 hour behind Banda, I guess. My name is Elsa. I'm the co-founder of Luminocean. Mareike is my co-founder as well. Together, we started Luminocean in November 2014. Back then, it was called Sea Ventures. Before we start the webinar, I want to share with you how we started it. It started when I was doing my sabbatical leave at the time in 2013. I met Mareike and Tuta in the Banda Islands. Tuta runs a scuba dive center, and Mareike was still doing her PhD in marine biology. I learned scuba diving from Tuta, and soon after, I fell in love with the underwater world, the island itself, and also the Bandanese people. The islands are really beautiful. People are amazingly friendly, and the underwater is out of the world. Really, really beautiful. For those of you who have come to Banda before, I'm sure you know what I mean. But for those of you who have not been there, you must go. I really recommend it since it is one of the top dive sites in the world. Lots of fish, big fish, small fish, turtles, and especially the beautiful coral reefs. If you see at the Google map, Banda Islands is pretty remote. It is located in the eastern part of Indonesia. It's really difficult to get there because of the lack of infrastructure, especially in transportation. Living there for the first 9 months in Banda and observing how the islanders live, how they do the fishing, how they handle their trash, especially the plastic trash, made me think that this beautiful ocean, the coral reefs, have to be protected. Otherwise, in a few years' time, we wouldn't be able to see them anymore. That's why Mareike and I started Luminocean, because we want to share with people about Banda Islands, about the ocean, not only the Bandas, actually. It's about the ocean in general. We want to create conversation. We want to create discussion, collaborations. Maybe many of you think, what can I do? I'm nobody, I'm not a scientist. I'm not a government officer. What can I do? That's why we are here. That's why we are Luminocean exists, because we want to connect people. Whether you're scuba divers, you're not scuba divers, you're scientists, conservationists, educators, or just common people like me. We want to create a conversation. We want to tell the public so that people understand, people can gain knowledge and then spread the words about how the damaging effects of overfishing, the pollution, and it's the effect to the coral reefs and to the ocean worldwide. And this is our first webinar with Mareike as a speaker. And we promise you that this will not be our last. So Marika, I hand over the screens to you. Best of luck.
Thank you.
Thank you.
Elsa already said a lot about Luminocean, and yeah, I want to say a few words about why we came up with this webinar. And actually, I hope that the Indonesian subtitles work well. Selamat malam juga ke semua di Banda atau Jakarta, Ambon, ya, di seluruh Indonesia. So I hope these subtitles are working. We selected English as the international language because we have people now joining from many different countries with many different languages. This morning I read in the news, actually another good reason for why we're doing this seminar and why we do what we do with Luminocean. I read in the news that most of the coral reef studies that are done in the world are not done by researchers from these countries where actually the coral reefs are, and that countries like Germany, like Canada, have a really large proportion of coral reef studies. So I think, what we want to do with Luminocean is create a network and connect more people from international research institutions with local people. So I think the most important part is the Sekolah Tinggi Hatta Sachir in Banda. And we want to motivate for research by Indonesians in Indonesia, and we want to help growing this network. So the other reason why we started this webinar is that everyone is kind of stuck at home now, and we already have this really nice network with Luminocean that we don't want to fall asleep, but we hope we can keep connecting with everyone who has joined us in the past, and maybe we can grow our network through this webinar. So it's a semi-scientific webinar, or in this webinar I'm going to present different scientific topics, but they're not going to be presented in a scientific way. I'm trying to do my best to present it in a way that it's easy to understand for everyone who's not from this field of biology or marine science. So also please interrupt me anytime if it goes too scientific, or leave comments and so on, and I hope I will improve then in the next sessions. So today I would like to start with the very beginning of where corals come from, so with the topic coral spawning, because it's a really fascinating topic. And this talk will be based on two scientific papers. The first one is multi-specific coral spawning events and extended breeding periods on an equatorial reef. And here, and this is about understanding when corals are spawning and how this is related to environmental factors, like what makes them spawn. And for the second one, I'm just gonna spend 3-4 minutes on the second paper. This is about a database that is growing, where anyone can enter data of observations of coral spawning. Yeah, coral spawning, what is that? We're talking about it all the time. It's the event when different coral colonies and individuals release their eggs and sperm into the ocean. And anyone who's not a diver or snorkeler maybe didn't have the chance yet to witness it, but maybe some of you have been so lucky to witness this at night. And corals are so-called broadcast spawners. Whenever you see this little shark swimming through, it means that it's some technical words that need probably explanation, and we are pasting the definition for these technical words into the chat, so you can see it, read it in the chat. So broadcast spawning means that eggs and sperm are released at the same time into the water column, and fertilization of the eggs happens in the water, not like in most animals on land that it happens internally, but it actually happens in the water. And this is the most common mode of reproduction in the sea. Most organisms in the sea use this technique of broadcast spawning. And what is spawning are the so-called coral polyps. They are the tiniest sections of a coral. So they almost look just like little circles, and each circle here is one coral cup where a polyp sits on, and you can see the bundles of eggs and sperms coming out. Or here you can see it even better, lots of egg and sperm bundles coming out. And if you look really closely, you see something that looks like tentacles. And this is the actual living organism inside these coral skeletons. These are the coral polyps. Coral polyps have a basic structure. They are so-called radial, or they have a radial symmetry. And many basic organisms have this. If you look at us humans, we have a symmetry, like you can basically divide us in half and one side looks more or less the same like the other side. But many basic organisms have so-called radial symmetry. That means if they look like a tube, they have one axis in the center and everything around is arranged symmetrically. So like a circle, basically. So these polyps are like little tubes with a body cavity in the middle, and on top they have many tentacles. These tentacles are used to catch their food, to catch plankton. And the skin of the tentacles is layered by different cells, and some of the most important cells are the so-called nematocysts. They are stinging cells. They have a little have some toxin inside them, and this is used to immobilize the plankton, to catch plankton, and then transport it into their mouth. So this is another typical part of basic organisms. They only have one body opening. Mouth and anus is the same. Yeah, everything goes in here and also goes out here. Corals also live together with symbionts, and these symbionts are called zooxanthellae. They also live inside the skin, and they help the polyps to survive by conducting photosynthesis. So they use water and carbon dioxide to produce sugars and oxygen. And for this, they need the sunlight. So they are photosynthetic organisms, same like trees that we have here on land. But they are actually tiny algae. And together with the polyp, they form a really efficient symbiosis and allow the coral reefs to grow to what they are. So these huge structures that you see could never ever grow like this without these tiny algae. And the polyp is actually what secretes these structures. The structures are also called skeletons, are made of limestone, calcium carbonate, and around themselves the polyps secrete a cup, also called a corallite. I think this is a really beautiful painting where you can see it much, much better. Normally you just see the surface, but below that you find lots of different channels and tubes that are all connected to each other. So the living tissue of the coral, when the polyps they live, they are on top of these colonies, and below that you find years, hundreds, thousands of years old structures, skeleton. And if the colony grows, this happens by so-called budding. So the polyp just splits off a small part of its body. It's a bud so a bud that comes from the parent polyp, and then this grows into a new polyp, and that's how corals grow. This can either happen extra or intra-tentacular. It's not really important to keep this word in mind, but just that you know two of the modes. So here on the left, you can see a case of extra-tentacular budding. That means the polyp, the bud, comes out from somewhere outside the tentacle circle. And in this case here on the right, it comes from inside the tentacle circle, so more like from inside the body cavity. And this results in really differently looking coral structures. So these beautiful structures and patterns that you see, they result from different modes of budding. And this growth of the colony, so this budding, this is actually asexual reproduction. It's a mode of reproduction, but not sexually, meaning the parent and its bud, they are genetically identical. So there are exceptions known already, but most of the polyps you find in this one colony are genetically identical. So today we want to focus a bit more on the other mode of reproduction, which is the sexual reproduction. And this is what you can observe when you observe coral spawning. I said before that most corals are broadcast spawners, where eggs are fertilized in the water columns. But about 1/3 of coral species are so-called brooders. So here, eggs are actually fertilized inside the colony, inside the polyps. And once the embryo is developed, then they grow into a larvae, and this larvae in corals, it's called planula larvae, gets released into the water surface. So this is the case for about 1/3 of all species. The larger proportion of species are the broadcast spawners, like what we saw in the beginning. So they release their eggs and sperms, embryos develop in the water column, and then the planula larvae resides. And this larva can stay in the water for 3 weeks till sometimes even a few months. And it spends its time being distributed by waves and currents until it finds a good place to settle. And if it finds a good place to settle, that's normally in the coral reef or near to other corals, on rocks, on hard substrates, then it becomes a so-called primary polyp, a young polyp. And this polyp will start to grow in the way that I explained before, so by budding, and it will form a juvenile coral colony. And this juvenile colony then grows into the adult colony when it can finally start sexual reproduction again. And this depends on species, but it can take between 2-3 until 10 years that a coral colony spawns for the first time. The larvae of brooders, they live much shorter. They only spend a few hours in the water until they start to settle down. Corals can either have separate sexes or can have both sexes at the same time. So they are either so-called gonochorists, which means male and female exist separately in different colonies or individuals. So here, as example, you find a Galaxia coral, that's a coral genus, which is only male, and then you have another colony which is only female. Now here for Fungia, these are the mushroom corals. They are actually, one polyp is what you see, so they're about this size when you see them in the water, And what you see is not a colony, it's actually one individual, one super large polyp. And they are also either male or female. So you can see either sperm or eggs coming out when they're spawning. But most of the coral species are hermaphrodites, which means that they have both sexes at the same time. And many of them have egg and sperm bundles. So the red dots here, they are not just eggs, they are actually bundles that contain egg and sperm. So when they are released, both is released into the open ocean. But normally they have something inside them that prevents them from fertilizing each other, so that this colony does not fertilize its own eggs, but it fertilizes the eggs of a different colony, and its eggs get fertilized by the sperm also from another colony, to make sure that there's more diversity in the genes of the next generation. Yeah, these are the basics of coral reproduction. So now we can start talking about the paper. So the next slides will be about this paper. And this is a paper from Palau. It was written by an international group of researchers from Palau, Australia, and Japan. And I selected this paper because it shows quite well which factors can be important to make corals spawn. And also it shows how coral spawning happens at a site that is not the Great Barrier Reef, because most of research on coral spawning so far was done in the Australian Great Barrier Reef. But this is a reef that is quite far south, so quite a high latitude for tropical reefs, which means there's much higher variation in day lengths. But most of coral reefs actually are located very close to the equator. So all the reefs in Indonesia, for example, and it's interesting to have a look at an example of coral spawning at a reef that is not so far from the equator. So the study was conducted at 2 different sites in Palau. Palau, you can see here, is between 7 and 8 degrees north. So in Indonesia, this would equal more or less the southern tip of Sulawesi till Flores, latitudinal-wise. So that would be 7 to 8 degrees south. In Banda, we are at about 3 degrees south, so we are even closer to the equator. And the authors selected many different species of corals at these 2 sites and were observing them for 2 years to find out when they are spawning. And I'm going to show you a little bit on which species they selected. But before we talk about that, let's talk a little bit about the classification in biology, because I'm sure not everyone is familiar with these words. As humans, we like to put everything into categories, into drawers. We like to know, okay, this belongs to category A, this belongs to category B. And that's also how this classification of life was done. So if you start at the highest level, there are the domains, and corals can be found in the domain of Eukarya. The other 2 domains would be bacteria and archaea. Then the next lower level would be kingdoms, and that's where you find animalia, so all animals. And within a kingdom, you find many different phyla. So the phylum of corals would be the Cnidaria. These are all organisms with radial symmetry that have stinging cells. This is jellyfish, for example. And then Below that is the level class, and corals belong to the Anthozoa, the flower animals, if you translate it, together with anemones, for example. And the level below that is the family, and here we will see many different families of corals. So the first group that they studied were in the family of Acroporidae and the genus Acropora. So the first group was defined as a coral genus of Acropora. And if you look at a scientific species name, you always see the genus in front, so in this case Acropora, and then you find the name of the species second. So in the study, the authors included 2 Acropora species, Acropora hyacinthus and Digitifera. And these 2 species are very common in Banda also. In the lava flow, you see lots of them, for example, for everyone who's familiar with Banda. Acropora is the most common coral genus. I think only at the lava flow there is already at least 30 different Acropora species. And you can identify them because they always have one central polyp or cup here at the tip. And then all the other corallites, polyps are surrounding it. And also the polyps always have 12 tentacles. So these are the keys to identify Acropora corals, if you want to look very closely when going diving again. Then the next group that the authors looked at was again a genus level, the genus Parietes. And here they just defined it as Parietes massive, and massive is not the species, it means it's this growth form, the shape of the coral. Massive means it's building a mound, it's building really a massive structure. And what Parietes corals have in common is that they have very tiny polyps. So you can see here the surface looks very smooth, almost soft if you look at it. Then the next group was only one species, but a very common species at this study site, again a Parietes species, Parietes rust, so which has a very different structure than the massive Parietes on the picture before. And because this is very common and the other one is also very common, they looked at it separately. And the next level was, or the next group was the Merolinidae. This is a family level. And the reason why here they chose the family was because they did not find so many different species there that they could observe for spawning. And they included 3 different species that you can see here: Paragoniastrea australensis, Platygyra lamellina, and Platygyra pini. So these have in common that they have these ridges on the colonies, really large polyps and ridges. And the last group they looked at were the Montiporas. This was again on genus level. Montipora is a very common genus also in reefs. In Banda we also see a lot of them. And they are often encrusting, so they're forming layers on rock and have also quite small polyps and often almost spiky looking surfaces. So here are the 4 examples of species that they looked at. But what did they do? They did something that as divers we would be painfully touched if we see someone doing that. They went to these colonies that they selected, and once per month they broke off a tiny piece of that colony. So many of them were branching corals where it was easy to break off a small piece, like a 4 centimeter piece they broke off. And yeah, then they looked inside and they checked whether they had mature or maturing oocytes. So oocytes are basically immature eggs, so that are eggs that are still developing. And if they could see oocytes, means eggs that are pigmented already, that have some color, they could see, okay, this colony is soon ready for spawning. And I did this once per month, always about 1 week before full moon, from January 2017 till September 2018. And if they found already maturing oocytes, then they started sampling the same colony every few days. And with this, they could then find not the exact day, but the month or week of when these corals released their eggs. So if they found oocytes on one day and not anymore the next time they sampled, they knew, okay, this coral had been spawning. So when looking at these different categories or groups of corals, they found that overall there was actually a really large spawning season. So looking at all of them together, there was spawning over 9 months in Palau. Whereas, for example, in the Great Barrier Reef, it's always considered to be really synchronized in 2 months of the year. But Acroporas had the peak spawning in March and April, which is similar to what you find in the Great Barrier Reef if you invert the season. So this would be springtime, basically. In the Great Barrier Reef, it would be October, November. And the massive Parietes were spawning or were peaking in April, May. The submassive or more structured Parietes Rus, was spawning from June till August, so completely different to the massive ones. And the Merolinide in September and October. And they found that Montipora had an extended spawning season over 9 months from February till October. So a very interesting and diverse pattern. And here's an example of the graphs for the Montipora species, where you can see how their results actually looked like. If you see a 1 here, it means there were ripe oocytes, mature oocytes, and the 0 means there were none. That means the time from 1 to 0 was the spawning time. And you see different individual colonies and when they spawn. So you can see that some colonies spawn twice per year, then not anymore in the next year. This is 2017, this is 2018. Then some spawned 3 times over the course of these 3 years, but not every colony spawned at the same time. And the same for Montipora undata. So there was also a very irregular pattern of spawning, but always between February and October, if you take them all together. So this doesn't look like a very synchronized spawning. But what they also wanted to know is how the spawning times that they found were related to environmental conditions. So they monitored 7 different environmental conditions. Here you see the sea surface temperatures, so the water temperatures at site 1 and at site 2. There was quite similar between the sites. They also looked at the daily light integral. And the daily light integral, it's a combination of the intensity of light. So was the sun very strong, sun very high at that time, and the day length. So the longer the day and the more intensive the sun, the higher the daily light integral. They also got data from weather stations about rainfall and looked at the tidal amplitude, so how low was low tide and how high was the high tide, because it was already assumed that spawning has to do with tides. They also looked at the atmospheric pressure and at sunset times, when did the sun set, and the day length, how long was the day. It looks like a huge variation here, but it's just a difference from 11.7 probably to 12.5 hours. So nothing compared to temperate regions. It's still a relatively low change in day lengths. So they did a statistical or mathematical model and looking at were the corals spawning, when were they spawning, and all these factors somehow related to it. And then they found out that there were two environmental variables that had no relation whatsoever. So rain didn't play any role, and atmospheric pressure didn't play any role in triggering corals to spawn. But all the other factors had an influence, but not on all species. For the Acropora species, it was the sea temperature that played a role, and this was the case for both species. So these two graphs are for Acropora, and the sea surface temperature and also the tides were influencing when these Acropora corals are spawning. And for Acropora, it was the time of the lowest low tide of the year when they started spawning, and a temperature of 29.4 degrees centigrade, which is a bit more than 1 degree less than the average temperature. That's what this means. So lowest tidal amplitude, lowest low tide, together with a certain temperature, triggered Acropora corals to spawn. And for the massive Parietes, it was completely different. Temperature didn't play any role, tide didn't play any role, but the daily light integral, so the available light, the amount and intensity of light that fell on the corals, was what triggered them to spawn. And for Parietes Rus, it was different again. So they didn't spawn depending on tide or temperature or light, but they were triggered by the sunset time. And this is, I think, very interesting from this paper to see that different species respond very differently, and you cannot automatically assume that everywhere in the world where you have coral reefs, all the species will spawn at the same time. So to summarize spawning in Palau, there was a long spawning season, an extended spawning season of 9 months in which different coral species spawned. But the most important season was March to May. It was 70% of all species that they recorded that spawned between March and May, with a peak in April. And the highest synchronization was in Acropora corals, so depending on species, up to 100% of all colonies that they looked at spawned at the same time in the same months. And the lowest synchronization was in Montipora corals. So they randomly, or what looks randomly to us, spawned over the course of 9 months. And they found that there were important environmental cues that played a role which was sea surface temperature, the daily light integral, the tides, and the sunset time. But very interesting, as I said, the different species or different taxa, different groups were triggered by different environmental cues. And these patterns that they found here in the study showed that it was very different in Palau to any other regions that had been studied in the past. For example, in Kenya, where you also have tropical coral reefs, there's a high asynchrony in spawning, especially also in Acropora corals, whereas in the Great Barrier Reef, there's an extremely high synchronization in spawning. And some studies were done in Yap and Guam, so Pacific islands, and there were more like spawning events of many different species that happened during always over the same 2 to 3 months, so quite synchronized also. And then, of course, the Great Barrier Reef with its mass spawning in October and November. And I think this shows how important it is to do more research on coral spawning, because knowing about it is very important for conservation. And it seems like that every site, or at least every region, needs to be studied. And if we want to know how should we protect corals in Banda, we cannot go and use data from Kenya or from the Great Barrier Reef. So all important regions with corals should have studies to find out when are the most important spawning times and when are which species spawning. And yeah, it's very important for conservation. If you look at these different environmental cues, how they are playing a role, and if you think about how water temperatures might change over time, can corals still spawn that synchronized? And coral reefs are stressed anyway already by bleaching, by sediment coming in from the sea, from overfishing. So if we now go and change, or not, of course not intentionally, but if these environmental cues change over time, and these are very important for triggering spawning, what will happen for reproduction in corals? So I think it's really important to know these cues, so we can know what we'll expect, and maybe we can help before it's too late. And yeah, that's why I think, especially in Indonesia, more and more research should be done on coral spawning. At this point, I want to talk a little bit about Andika, who's also here. Maybe Andika can say hi later and answer more questions. So Andika by that time was a student from Bogor University, and he researched corals in Banda. When were they spawning in Banda? And most of the nice videos and pictures I have of spawning corals from Banda resulted from the project for his work. So we supported him with Luminocean to do his research. And why I want to emphasize his work here is because he used a completely different technique to find out when corals are spawning than we have seen in the paper. You saw like they needed to break off some pieces of coral to find out if they were spawning or not. And Andika installed egg traps to find out if a coral was spawning the previous night or not. So you see it on the picture. They are very simple. Price of one trap, I would say €3. A low cost technique and least invasive technique. So it's just an upside down funnel with a bottle attached to the top. And if this funnel is attached to the coral colony, it will catch some, not all, but some of the eggs and sperm released. So if you go back and look at it the next day, you can see if it spawned or not. The only disadvantage might be that you have to go very often, so you need more resources on diving tanks and so on. It's a lot of work, but at least it's a very low invasive technique to check for spawning. And what Andika found in Banda was, he identified different species that spawned, and he actually identified them with genetic tools by extracting the coral's DNA from the eggs. So also no need to break off anything. And then he found that there were 2 main spawning events in Banda, which were in October, November, and in March, April. But some corals also spawned in December, like Pectinia. So in Banda, there might be also an extended spawning season, and it's still a lot left to find out about whether same colonies also spawn twice in a year, or whether there's this really peak of a mass spawning in October, November, or again in March, April. So maybe, this can be also an idea for students from Hatta Syahrir to continue with this research. And with our scholarship program from Luminocean, which will hopefully soon start again when I'm back and COVID-19 is not a problem anymore, that we can train more students in diving and make it possible to follow up with these projects. Rafa is training a lot already to dive now, so maybe also some students, and maybe there will be more future researchers from Banda. I just want to spend a few minutes on the second paper. This is just a hint, and it's a paper that Joanna Diaz found and sent me. And this is an open access spawning database that is growing now. So this is a chance for anyone who observes that corals are spawning to contact the main author, Andrew Baird, and add all the observational data. And you don't need to be a scientist to do that. You will get help with species identification. If you have a picture, if you have a video, and you have recorded the time when spawning happened, then you can contribute to this database. And this is what is in there so far. Lots of researchers just joined together and started with the first data sets, and now it's open for everyone to contribute. So these are between 10 and hundreds of spawning records per site that are included. So this will help a lot to find global patterns of when corals are spawning. So it will be huge for understanding more about this topic. You can see there's still no data from Banda, so I think would be really nice for Bandanese researchers to contribute to this database. It's just an example of how the data input can look like. It always starts with the site and depth and time to prevent double entries. If 2 divers were at the same spot at the same time, this database will make sure that only one time is recorded, that no colony is recorded twice. And there are lots of additional details, but anyone who's interested in contributing will get proper instructions, and these instructions can also be found in the paper that I cited. I will put the paper in the chat. Yeah, so I think what we can learn from these papers is maybe some motivation of going diving again. I don't think anyone needs that extra motivation, but also of keeping our eyes more open for what's going on underwater, and maybe networking more, collaborating more, and whenever we find these interesting observations, really finding a way of contributing them to the knowledge base. And maybe that's one advantage of COVID-19, that all these online things are becoming more common for everyone. And I think we will soon see more online databases like this corresponding database, where divers can really contribute to topics. And there's so much valuable information that's seen by divers, which is just lost out there. So I hope that maybe also with Luminocean we will start some type of database at some point, and I hope everyone can contribute a little bit with having fun when diving and adding what they see. So I hope you enjoyed, and I will open up for discussions and for questions, and if you have suggestions for improvement for the next webinars, please let me know through chat or through email. Thank you.